What is life? These three simple words spun out a universe of questions that is no less easier than these words themselves. What is life? How do we separate the animate from the inanimate? What are the basic ingredients of life? Where did life first stir? How did the first organisms evolve? Is there life everywhere? And to what extent is life scattered across the cosmos? These questions, complicated yet unanswered, outlines the framework for the subject biology. Now, biology, this word literally means the study of life, and the whole history of biology from the ancient days where Aristotle, father of biology and another bunch of cool subjects, first proposed the classification system, to modern days where we deal with computer-brain interfaces and selective organ targeting and another bunch of cool words like that, is really but a history and an attempt to define life, and if we be a bit more ambitious, perhaps to grasp life. And so here, I would call traditional biology the study to define and understand life. We look at different parts of life in an attempt to understand them. We look at cells, the basic building blocks of life. We look at genes, the information encoded in life. We look at chemical reactions, the driving forces of life. By looking at all these components of life, we try to grasp and piece together our answer of what is life. So we've been doing this process for a few hundred years, a few thousand years, and it should be recognized that we've made progress at an alarming speed. We're really beginning to understand what is this concept of life. But traditional biology is slowly reaching a plateau phase. You can probably tell by the rate of unemployment of bio undergrads. It's ranked first in all of these. Therefore, to fully understand life, we'll need some new methodology. Now, one of the first underlying principles of engineering is, if you don't understand something, try to break it down into its smaller components. Look at each component separately, maybe glue some components together and see what you get. And right now, every subject is undergoing the transformation of becoming digital, where with the aid of computers, they made astronomical calculations possible and helped us break through the limitation of the human mind. So modern biologists introduced engineering and computer science into traditional biology. And right now, instead of directly trying to define life, we now approach the study of life through a new way. We now try to morph life. So, the word morph is actually used quite frequently in the context of biology. Morph itself could be directly used to mean a local population of species consists of interbreeding organisms and is distinguishable from other populations. Morphology is a branch of biology that deals with the form and structures of plants and animals. And the word morph can also be placed in metamorphosis, one of the most favored words of elementary students in China, because metamorphosis has an interesting translation in Chinese. The word itself literally means the transformation from caterpillars to butterflies, but here we go. 变态 as in 变化形态, metamorphosis. Whatever the thing is, the word morph, we can see that the underlying idea is a change in the form of something. And in the context of biology, we're changing the form of life. Okay, here comes my favorite part. It's this part where every time I talk about modern biology, people just give me this look that you would give to those, evil scientists that try to make mutant pig meat take over the world. The thing is, we're actually doing that sort of stuff. Okay guys, let me explain. For starters, we're not evil scientists, and we're not making mutant pig meat. We're making mutant chicken meat. It does make a difference, and we're not using these meat to take over the world. This is actually a brand new field of study called cultured meat, where we use stem cells and cultivate them in vitro to develop into meat tissue. Sounds very fancy, huh? But basically what we do is, we take a bunch of magical cells called stem cells that has the capacity to develop into any tissue in an organism. We put them in a lab, and we make them develop into chicken meat, which we want. It's actually meat grown from meat. So, cultured meat doesn't require animals to be slaughtered the way traditional meat does, while producing meat that is less contaminated, contains fewer antibiotics, while at the same time being as tasty, as juicy as meat just cut off from chicken. So stem cell engineering, this broader field of research, aims to utilize the amazing regeneration and morphing ability of these stem cells to their fullest extent. And I don't know if any one of you here pays attention to the Nobel Prizes. Just 3 years ago, in 2020, the Nobel Prize in Chemistry was awarded to 2 female scientists, Emmanuelle Charpentier and Jennifer A. Doudna, for the development of a method for genome editing, the CRISPR-Cas9 system. Although for some reason this prize is given to the field of chemistry, which my chemistry teacher has complained several times with us, this is definitely a revolutionary breakthrough in biology. So we can basically understand the CRISPR-Cas9 system as genetic scissors. In simpler terms, what the CRISPR-Cas9 system does is it locates a piece of a gene precisely in a cell, cut that piece off, and if necessary, we can put a new piece of gene inside. So such techniques that deals with the modification of genes is what we call genetic engineering. And genetic engineering is incredibly influential and revolutionary to modern biology. So what exactly do we use this magical technique for? Well, we get to create bacteria that glow in the dark. Carousel that turn pink. I mean, it's pink. Okay. On a practical level, we can use genetic engineering to create crops that are resistant to pest infection. We can create hepatitis B vaccines at an unimaginably low price. And we can also create CAR T cells that precisely eliminates cancer without harming any of the good cells. So genetic engineering, with the help of genetic engineering, we are not just dealing with life forms that naturally existed in the wild, but we are changing life, shaping life into whatever form that we wish. Now we're beginning to morph life. And a series of questions I proposed at the start of the speech, the questions encompassing what life is, come from one of my favorite books, Life at the Speed of Light. The author of the book, J. Craig Venter, revealed in his book a revolutionary project and perhaps the whole history of biology and science, Project Cynthia, Syn for Synthetic Life. So basically, exactly 13 years ago, in July the 2nd of 2010, Venter published his results on Science, where he led a team of researchers in an attempt to synthesize life from nothing. What he does is, so we know that a cell is controlled by the genes inside it. The genes are built from little building blocks we call nucleotides, and the collection of all genes inside a cell is called its genome. So the researchers designed a simplified version of the genome of a bacteria called M. mycoid. They synthesized this genome from basic nucleotides using computers to modify and decide what genes to put inside the genome. They inserted the genome inside an empty cell, and the empty cell began to behave exactly like a real living cell. In Venter's words, what they have created is the very first self-replicating species on the planet whose parent is a computer. So here we are with the field of synthetic biology. What we want in synthetic biology isn't just to cut off a gene and then insert a new gene, but to design new biological parts, devices, systems, and ultimately design life from scratch. Although Cynthia is probably not, debatably not the first life from scratch because her genome is still based on a real organism, she is definitely one of the first solid attempts we have had on morphing life. So if you think about it, modern biology is really advancing at an incredible speed. 100 years ago, we're still having a heated discussion about what is the shape of DNA, and 100 years later, here we are with genetically modified immune cells with chimeric antigen receptor immunotherapy and combination therapy. Whatever in the world that is. But as we morph life, as we change the form of life, as we let imagination and creativity be the only barrier to what life forms we may create, it's natural to ask the inevitable question. How far are we from the future outlined in science fiction and dystopian novels, where humans create a life form that we are no longer able to control, or that we begin to modify our own species, turning ourselves into digital life or cyberpunks that eventually led to our own downfall? Do we still have another 100 years, another 13 years, or another revolution of the sun? And who's to say that we as humans have the right to morph life, to shape life into whatever form we want? That we're the gods of this earth, instead of mere mortals with such self-assured hubris and audacity that we allow ourselves to play the role of God. That we're not Icarus, who's flown too close to the sun. Fortunately, just right now, I can say there's still some distance between the future outlined in dystopian novels and our reality right now. Right now, the majority of works that biologists in the field of genetic engineering do still involves picking a gene that encodes an interesting protein, putting the interesting gene inside an E. coli and forcing the poor E. coli to produce that protein for us. So really, there's still no need to worry about mutant spiders designed by evil scientists taking over the world in the foreseeable future. And I apologize in advance for anyone who's thrilled to become Spider-Man, bitten by a genetically modified spider after my speech. Really, if you ask me, mutant E. coli taking over the world is a much greater possibility than spiders or Frankenstein or zombies. But when we chill down and think about it, the future is not that far away from us. In 2018, Professor He Jiankui from China announced to the world that the first genetically modified human babies were born, the CRISPR babies. He edited the CCR5 gene of 2 babies while they were still embryos, making them naturally resistant to AIDS, but also making them born into a life full of uncertainties and dangers. The thing is, we just don't know what will happen to these 2 babies. The news sent shockwaves around the world as people were appalled by his actions of modifying the genes of human infants. He Jiankui himself was also sentenced to 3 years in prison in China. But interestingly, he was also awarded Nature's Top 10 People Who Matter this year in 2018 and Time's 100 the most influential people in 2019. Influential. He Jiankui was really influential in a sense, for he reminded us that playing with the genes of human beings, modifying the genes of infants, is already a reality 5 years ago. And from the speed of biological developments, like every day there's new breakthroughs in biology, we will probably be at Frankenstein by tomorrow noon if nothing happens. So really, what sets the boundary between us and the future outlined in dystopian novels? I would say it's morality. Without morality, things would be good. Without morality, if we say that it's okay to do experiment on people or life doesn't matter because everything is for science, biology would advance at a much greater speed. In fact, I would say that considering that human nature is just brave and curious and daring, CRISPR babies would have probably existed back in 2010, and standing here making the speech, giving a speech to you will probably be a cloned version of me, who has already a ton of gene edits. Without morality, without the limitation of morality, biology would advance at a much greater speed, and the possibilities of morphing life will be truly limitless. But think about the future again. We have to remember that biology is not just the subject of life, but one for life. Life and the respect of life are a pair of topics incorporated in biology that are naturally inseparable and cannot be ignored. Because if we ignore life, if we decide not to respect life, if we pursue science for its own sake, what is to become of our species? Thank you.